All right, so we've got about 20 slides here that are different tissues from around the body. And what I've done is put them in here as a duplicate. So there's 20 slides with no narration, and then the last 20 slides will have narration. And what I'm going to do is kind of walk you through how to tell which tissue is which, like I did in the histology lecture. So my recommendation is to do this first part a couple of times. See what tissues you think are indicated, and then that way you can get an idea of where you're at. I would not recommend listening to the version that I narrate right off the bat, because you really want to see if you've gotten the material yet, and you're not going to have a chance to do that if you're already knowing what the answers are. So from here on out, I've got animations on each slide. And what will happen is you'll advance to the next slide, and then as soon as you click on the screen, it'll start playing the animation, and there should be arrows and figures that are timed to appear with that. But you have to click on the screen once to play those, and then once they're done, if you click on the screen again, it'll advance to the next slide. One thing I'd like to point out is that histology is as much an art as it is a science. And so the variation in techniques from how slides are made, variation in skill from the people preparing them, how long they've been around, all of those influence the color and how clear certain structures are. I've tried to pick good representative slides of each tissue, but you may have access to different ones through your textbooks, through your lab manuals, or through your courses when we do go back to on-site delivery. And so try to look at these as not memorizing the image, but understanding the pattern of the cells and extracellular matrix and free spaces and organization that we see in these slides. And to really get a good handle on that, you really do have to look at a lot of slides. There's no easy way around it. But again, I really don't recommend memorizing slides. Try and understand why they are what they are. All right, so the first thing that we do when we're trying to figure out what we've got on this slide is we need to figure out the primary tissue classification. So the primary tissue classification in this is most easily determined because we have a very large free space that's visible on here, which tells me it's an epithelial tissue, the lumen of this pathway. Now that also tells me, since it's a lumen, it's not just like an open border, that I'm in a duct of some kind. And that duct then is most likely going to be a mucous epithelium. That's also indicated by that kind of purplish, more solid layer that we see on top of the epithelial tissue, or towards the apical surface of the epithelial tissue. Now that I've figured out that's a primary tissue type is an epithelium, it's likely in a mucous epithelium, then that means there's only a couple of options that it could be. That lumen being kind of star-shaped the way that it is indicates that this is probably in the urinary tract or urogenital tract in a female or male. And so knowing that, and knowing that it's an epithelium, we need to take a look at the top layer of cells and figure out what's going on. So the top layer of cells, which I pointed out with a few arrows, that is going to be a it's not really squamous, it's not really cuboidal, definitely not columnar, so that's making me think it's transitional. And then there's a few cells which had the arrows on them that have 2 nuclei in that top layer, which is a definite giveaway that it's a transitional epithelium. All right, on this slide, we need to start off just like the last slide by figuring out what our primary tissue type is. And in this one, we do see a little bit of a white space. However, that's not a lumen. And this is a good point or moment to point out that sometimes you can get spaces or white edges in the slides because of the histological technique, not because it's normally there in real life, or I should say in vivo in the actual organism. And in this case, that white space is just some of the cells separated apart. So how do we tell then what primary tissue type do we have? Well, it's very cellular. I see a lot of nuclei. They're kind of pinched into long rows. And I also see a very clear, faint striated, like dashing along the cells. That perpendicular dashing or striation to the long axis of the cells tells me that this is most likely striated muscle, which means it could be either skeletal muscle or cardiac muscle. And the way to tell the difference between the two is to look at the nuclei. Now we can see that it's multinucleate. There's many nuclei per cell, and I also don't see any intercalated discs, which tells me that this is going to be skeletal muscle. On this slide, we see right off the bat that there's a number of clear white spaces, and that those spaces are not linked to one another. Each one is fully encapsulated by a membrane of some kind. That rules out the possibility of this being the lungs. And when you see this many spaces, there's not a lot of extracellular matrix, and we can say that we're not in the lungs. That only leaves one option. The primary tissue type would be connective, and the specific tissue type would be adipose. Now in this slide we've got another tissue to take a look at, and the first thing that I notice is on the top portion of the slide we have a clear apical surface with an open space or a free surface there. That tells me right off the bat we're dealing with another epithelium here. So our primary classification is epithelial tissue. Now I look down to find the basement membrane, and I've got to go quite a ways down to the bottom of that bracket, and that indicates that our basement membrane is pretty far away from the apical surface with a lot of cells in between. So we've got a stratified epithelium for sure here. Now to identify the epithelial type, we need to look at the shape of the cells at the apical surface or near it. Now what happens in these big stratified tissues is the apical cells are dead. And so sometimes they lose their nuclei, sometimes they get a little beat up. So we may need to go a couple layers down, and then at that point we can see that these cells are stretched out, so they're going to be squamous cells. And so our tissue type here is stratified squamous epithelium. This is non-keratinizing. I don't see that like fibery apical layer on there, so that means that this must come from the vagina, anal canal, esophagus, or the oral cavity. Here we do not see any apical surface. All the borders of the image are leading now to just more tissue. So right off the bat, you can say that there is no epithelial tissue in this view, which limits our new options now to nervous, connective, or muscle. Now there's kind of a lot of nuclei in here. However, this is spread out pretty thin, so those nuclei have a lot of space in between them. That means that it's probably not muscle, leaving us with nervous or with connective tissue. Now I don't see any big neurons, so that indicates that it's a connective tissue. But what kind of connective tissue is it? When we look a little bit closer, I can see those big pink ghost-like strands. Well, those are going to be our collagen fibers. Our nuclei then mean that those are fibroblasts. There's going to be a few other cell types scattered through there, but we only need to identify fibroblasts in this. And then the thin, dark, hair-like strands are elastic tissue. The presence of these fibers in these cells arranged in this way indicates that this is going to be areolar connective tissues. This is that one that's found in the lamina propria of pretty much underneath any epithelium, and provides a nice sort of like cushioning, generic connective tissue for structures. All right, now the brackets on this slide indicate that we're looking at the bulk of the tissue. If you do look at the extreme right corner, upper corner, you'll see a free space, and if we were to zoom in, we would see a simple squamous epithelium. However, the rest of the tissue is not epithelial tissue. And we can look at that and we see it has a lot of nuclei, those purple dots. And so that's going to tell me that this is most likely muscle or epithelial, which we ruled out epithelial. So we're probably looking at muscle. So is it smooth, skeletal, or cardiac? Well, there's no striations, which is a dead giveaway that we're looking at smooth. We can also verify that by looking at the arrangement of the cells. In the small intestine, they're usually arranged where a layer will wrap around the intestine itself, almost like a watch band around your wrist, and then an outer layer will run down the length of the intestine like your sleeve runs down your arm. And that's what we're seeing, and I've indicated the difference between the two layers of that dashed line. Now this slide has some clear rings or circular structures. It looks like we're looking from the top down on a forest that's been clear cut. That's a giveaway that we are looking at bone tissue. And so this is where the framing of the question is very important. If you're asked what the primary tissue type is, it's connective tissue. If, however, you're asked what the specific tissue type is, it's going to be bone. This slide looks a lot like our smooth muscle slide that we had a few slides ago. But there's some key differences in this. One, our fibers are arranged in parallel rows, and the nuclei are squished up little thin lines that are kind of smashed between those rows, almost like skeletal muscles nuclei were smashed between the cells. However, it's not striated, so it's not a muscle. So when there's a lot of tissue here, the pink, however, it appears to be extracellular. So that's making me think connective tissue is the primary tissue type. Seeing the parallel arrangement of fibers that run left to right on the screen, that indicates that this is going to be dense, regular connective tissue. So this is likely from a tendon or a ligament in the body. This side depicts a central mass of darkish staining tissue surrounded by two clearly connective tissue in origin bands. Right off the bat, I'm realizing I say that all the time now, sorry for that, is a perichondrium. So that's what those two layers of connective tissue on either side is, which means that the main mass in the middle is connective tissue also. And since we can see a perichondrium and those look to be a lot of lacunae that are densely packed and it stains very dark, that rules us down to a cartilage and specifically elastic cartilage. And if I zoom in, you can see the dark fibers that are contained within here. Now I have this image zoomed out a little bit so that we could get the brackets on there and it could you could see it easier. But you could still see the lacunae, the chondrocytes, and the elastic fibers even at this less magnified view. This slide should look familiar because it's a similar tissue type to what we've already seen. Now there's two tissues that we can identify on this one, really three, but I've just indicated two. Now point A is indicative of an epithelial layer, and this is actually a great demonstration of how when you make a slide you can actually cause some damage to the tissues. So about 2/3 up the epithelial tissue you see a big separation between the cells. Now that's not natural, that's because of how they did this slide technique. Well, we do see the true apical surface at the top of the image. Now, given how thick and cellular that epithelial layer is, that is definitely going to be a stratified squamous epithelium, even without looking at the cells themselves. Likely, that thick layer will also be a keratinized stratified squamous epithelium. And this is something we haven't talked about yet, but the reason it doesn't look hairy or have a lot of air spaces like we saw before is this comes from either the palm of your foot or your fingertips, sole of your foot or the palm of your hand or fingertips, where you've got thick keratinized stratified squamous epithelium. You'll need to know that for the integument system, but not quite right yet. As we go down towards the lower edge of that bracket, you'll see the basement membrane, and then that's a very kind of twisty, curvy basement membrane that runs across the screen. Again, another indicator that you're looking at something from the hands or the feet. Now, point B is lighter staining, there's fewer cells, and it's deep to an epithelium, so it's most likely a connective tissue. Those big pink strands that move through there are not doing so in an organized fashion. So I could identify this on two factors really alone. One, just knowing that those collagen cables are routing through in a really disorganized way tells me that it's dense irregular connective tissue. The other thing is knowing that underneath our integument, underneath our skin, you will find a layer of areolar connective tissue. That's the gap between A and B. And then beneath that is always dense irregular connective tissue. So you could get that answer from either knowing the structure of the skin or identifying the collagen fibers themselves. Now, this slide has a couple different tissues on it. However, our arrows tell us what's being asked. Now, the heads of the arrows fall into a lumen, into a free space, so that tells me where the apical surface is. And actually, for both rows of cells, you can see that they share that same apical surface. In your small intestine, there are finger-like projections called villi that are coated in an epithelium that help you absorb food. So that's what we're looking at here, is the gap between two villi. If you hold your two fingers up in like a V, then the surface of your skin that's facing each finger would be what we're seeing here. Now, the actual type of epithelial tissue we can tell by looking at the nuclei and the cells. So if I look at the nuclei, they're all arranged in one row, so that indicates it's a simple epithelium. They're oval and they're polarized. That is, that they're closer to the basement side than they are to the apical side. That tells me that they're columnar. So we have a simple columnar epithelium on this slide. Similar to the last slide, we have a longitudinal section of something that is long and cylindrical, and that there's going to be free spaces in it. Now, we can only see one free space where the arrows are at in this image, but if we were to expand this view, you'd probably see some more. So, if there's a free space, then that tells us that we are looking at an epithelium. At least that's what's indicated on this one. Again, there's a couple tissue types here. But the indicated tissue is an epithelial tissue. And if we look up close, we see the nuclei are in one row. So that's going to be a simple epithelium, and that then the nuclei are round and they're roughly in the center of the cell. So that tells me it's simple cuboidal epithelium. Hopefully you're starting to get the hang of this now. So what we have here is another tissue that has a perichondrion. So two layers of kind of, it really looks like a denser regular connective tissue, those thin layers by each edge of the bracket. It's surrounded by other connective tissue. All cartilage will be surrounded by other connective tissue. That perichondrion told me it was cartilage, plus we see that the matrix is got lacunae with chondrocytes in them. Now this is a little bit weird because you can see that the cells are actually, or the lacunae are pretty dense, which would make me think elastic cartilage. However, I don't see any black fibers in the actual matrix of the cartilage itself, and so that rules out elastic cartilage. Fibrocartilage does not have a perichondrium, so it can't be fibrocartilage. Plus fibrocartilage, you see these like fibers, the collagen fibers routing through the matrix. So that means that this is hyaline cartilage. Because of the shape of this, you can also tell that it's actually from the trachea, and so this is part of the respiratory tract. And if you zoomed out a little bit, you would see pseudostratified columnar epithelium nearby it. This is a slide from the kidney, And our arrows are telling us what specific part to look at. Again, there's multiple tissue types on this one, so you always have to pay attention to what you're being asked. Now, those arrows are pointing directly at the nuclei of a tissue that we need to identify. The arrows are in a free space, they're in a lumen, so that means that we're looking at another epithelial tissue. Now, those nuclei look stretched out and oval-ish, and instead of a big cell, their cell is so narrow that really all we're seeing is the two edges of the membrane as it wraps around that cavity, the cell membrane, that is. And so that tells me right off the bat that this, I did it again, that this is a simple squamous epithelium. If you want to look up more on this particular one, this is a Bowman's capsule that we find inside the kidney. The kidney actually has a bunch of epithelial types in it, so it's a great one to look for when you do get back to microscopes. One thing that should stand out to you the first time that you look at this image is that we have a lot of dark or sepia colored stain here. That indicates that we're looking at something that has been stained with a silver stain, and that stain is most frequently used with lymphatic tissues such as lymph nodes and the spleen. That can rule down your options at the beginning. I almost said right off the bat, but that tells me that I'm probably looking at a connective tissue and specifically reticular tissue. And then when I look at the actual fibers themselves, it is reticular, and you see it looks almost like tree roots with that branching and kind of curvy shape to it. There are a lot of cells in here. You've got to look through the reticulum to see the nuclei, but there are a lot of cells in here. So this is likely from a lymph node or from the spleen or from a tonsil, somewhere that has that lymphatic tissue. So the primary tissue type is connective tissue, specific is reticular connective tissue. Hopefully you were able to pick out the lumen or the free surface on this already on your own. And then you can see the arrows there are indicating a thin layer of cells. Now, when we look at this, the cells are so thin that it's really hard to even see them, which is a really obvious indication that we're looking at simple squamous epithelium. Now, the one thing that you could do is just miss that there's a layer there even and start looking at the massive cells beneath it, which are also very cellular, which indicates either an epithelium or a muscle tissue. However, those cells underneath it, you'll see that there's only scattered nuclei, and then there's also cylinders of lighter pink, meaning that we're getting cross-sections through cells, sometimes hitting the nucleus, sometimes not. So that area underneath is smooth muscle. This is actually from the smooth muscle slide, and we've zoomed in on that serosal membrane that's on the periphery of the intestines. All right, now striations leap right off the page at me as soon as I look at this. And so that indicates that I'm looking at one of two tissue types. It's going to be a primary type of muscle, and it can either be skeletal or cardiac muscle. Now I can tell the difference between skeletal or cardiac very easily in this slide. One, there's not multinucleate cells. The nuclei aren't crowded along a single line. The cells do seem to branch in Y shapes as opposed to being one long cylinder. And then the other obvious giveaway is that we have intercalated discs, which indicates that it can only be cardiac muscle. Here we have a nervous tissue slide, and we don't need to go into the specific tissue types because we don't really do that. Later on we'll talk about the different kinds of cells that we encounter in nervous tissue, but for right now you really just need to know it's nervous tissue. That should be clear from the big neuron that's right in the center of the slide. That looks a bit like an octopus or a squid with all those tentacles coming out. Then within that neuron, usually you have a pretty dark staining nucleus. And if you were to zoom in, you probably see either a darker spot or a lighter spot within the nucleus that would be the nucleolus, but it's really small, so you'd have to zoom in a lot. The other nuclei that we have scattered around are neuroglia, or also they're called glial cells. There's far more of them in nervous tissue than there are neurons, but they're going to support that neuron and the surrounding neurons. All right, hopefully the free surface was clear to you. Our arrows are in that free surface indicating the apical side of an epithelial layer. Now I need to figure out which kind of epithelium it is, and there's a few key identifiers in here. One, the cilia. Those little hair-like extensions, they're there to help move mucus across the surface. That really indicates that we're in the respiratory tract, most likely. So that puts me right off the bat thinking that's a pseudostratified columnar epithelium. I can confirm that by looking at the nuclei. While it might appear as if there are two layers, there actually aren't. Those cells are all touching the basement membrane, and most of them reach the surface, but there's a lot of cells that are kind of crowded out from the surface. However, they're all still touching the basement membrane, so they're all one layer. The other thing is the goblet cell that appears in that layer too. Those are frequently found in the respiratory epithelium or pseudostratified columnar epithelium. They're also found in simple columnar, but this is clearly not simple columnar epithelium. All right, our last slide for this practice quiz or demonstration or study tool, however you want to think about it, is a blue staining tissue. That indicates that it probably was a Mason stain that was used on this, which means it's probably connective tissue. And if I look closely at the cells, I can see that there's actually a little kind of ghost-like hole that they're within. So that's most likely a lacuna. Now I say most likely in all of these because what you're really doing is trying to rule out other options. And this is one of my least favorite of all of the connective tissues, really all the tissues. This is fibrocartilage. I can tell that because of those lacunae, which are, a little hard to spot at first, but once you see one, you should hopefully start to see a lot of them. And also I can see these bundles of collagen that are almost parallel but not quite in a otherwise smooth, glassy extracellular matrix. Now these can be zoomed in as is here. This is found at the end of a long growing bone, or they could be a little bit more zoomed out on a vertebral disc where you would see that it's actually those fibers are forming like a circle to help resist that compressional force. But the main points here are that we have chondrocytes and lacunae, and we also can see these collagen fibers that are not elastic, so we know it's not an elastic cartilage. That rules out hyaline, it rules out elastic, leaving only fibrocartilage as our option.